Okay, good day to everyone. I'm Keat Lay. I'm the Field Application Specialist in Apical Scientific. So welcome and thank you for participating in today's webinar with the title Give Your PCR a Hug. Okay, so when I go through the slide, if you have any questions, do write in the chat box. I will try my best to answer you after the slide sharing. But if I can't answer it or I may need to get more information to answer you, I will certainly get back to you through email later. Okay. So throughout this session, I will ask 3 questions. So anyone who give the answer the fastest and correct, you will get a mystery gift from us. Okay. And every question will pick 2 winners. So do write out your questions in the chat box and follow with your registration name, okay, to answer it. Okay, so not to delay, let us continue to the content today. Okay, so in this webinar, we'll go through a little bit on introduction on PCR and why polymerase is a hard for your PCR. And then we'll talk about the 4 key features for PCR study. After that, we'll go into more detail on how to choose the suitable polymerase for your PCR with the help of Thermoscientific polymerase and their family members. And lastly, we will share some tips and tricks for you to better success in your PCR. Okay, introduction. Let's start to know a little bit on the history of PCR. The story starts in 1970, where the single-stranded DNA replication was reported, which involved the use of primers and DNA polymerase. Then the tag polymerase was isolated from bacteria, Thermus aquaticus, which live in a high heat place. So the polymerase will not destroy in the high temperature which used to denature the DNA. Okay. After this, the PCR technique was invented by Carey B. Mullis, who determined his Nobel Prize at 1993. From here, it actually started the very active of PCR development, where the PCR machine was invented. Then later, the first Pcr Kit was developed. Okay, so from here, from this history, you can see how Pcr start. It's actually start when there is a DNA polymerase was found, right? So this is the reason we say that polymerase is actually the heart of the PCR reaction. Just like a baby, you can see it start with a heart. When the heart start bouncing, so the baby start to develop. Okay, so this is why polymerase is the heart of the PCR reaction. Okay, but since 1983, where PCR was invented, upon these 39 years, researchers often encounter problems by using PCR to get what they want. Here are the 5 common PCR complications which researchers facing within upon these 39 years. Okay, so the difficulty in amplify GC-rich template and also long template is one of the difficulty. Then the determination of non-specific products in their PCR and also errors found in the amplicon. Also difficulty in amplify the sample which carry inhibitor. Okay, all these 5 are the common PCR complication that is faced. Now with the advancement of technology, researchers found solution for all these complications. It is either with use of polymerase other than tag polymerase, for example using PFU DNA polymerase, or improve the reaction buffer to increase the activity of polymerase, or use PCR enhancer to tackle inhibitor in the PCR so that the polymerase can act better in the reaction. And the last one is use a modified DNA polymerase with desired properties like fusion DNA polymerase, so it helps to enhance the PCR. So from these two slides that I mentioned until now, we can see that PCR start after DNA polymerase was found, and also to tackle the problem encountered in PCR, researchers actually focus to enhance the activity of polymerase also. Okay, so like what I mentioned, PCR need polymerase, and polymerase is the heart of the PCR. Now, let's understand more about this DNA polymerase. Okay, so DNA polymerase play a crucial role in DNA replication and DNA repair inside our cell, okay, in vivo. Also, it is used in molecular biology application, especially in PCR and sequencing. DNA polymerase have two activities. One is the 5' to 3' polymerase activity, where it will add the NTPs to the 3' ends of the extending DNA fragments, and then which is from 5' to 3'. Okay, that's why it's a 5' to 3' polymerase activity. And another one is the 3' to 5' exonuclease activity. Okay, which this is actually the proofreading function of the polymerase. It is like our auto correction when we typing using our phone. Then it, when the polymerase found there is mismatch in the extending fragment, it will go back automatically and then correct it. Okay. Okay, now we need to know what is the PCR features, what is the key PCR features that you are looking for before you can decide to choose which polymerase to be used. Okay, so there are actually 4 key features for PCR specifically. Okay, so there is there is specificity, accuracy, special PCR, and also fast workflow. Okay, I will explain it in detail in the next few slides. Okay, so first we start with specificity in PCR. Okay, traditionally during the reaction setup of PCR, primer can bind unspecifically to the DNA template or to each other. So this misprimed DNA duplex can be extended by DNA polymerase. So if you are using a proofreading enzyme, the primer can degrade due to the 3' to 5' exonuclease activity of the polymerase. So all this scenario can result in non-specific amplification in a PCR. We have the hot start technology, which can help to solve all these difficulties in the PCR. Hot start polymerase is a type of modified DNA polymerase, which carrying a mixture of tag DNA polymerase with an inhibitor, which can be either an antibody or antibody or chemical modification or an adapter. Okay, this inhibitor will bind to the polymerase and inhibit the activity when the temperature is below 45 degrees Celsius at the room temp. Okay, so when the temperature go up in a PCR to a normal cycling condition like 50 something or 60 something, the polymerase will release and then the amplification will only start. Okay, This actually allows you to prepare your PCR mixture at room temperature rather than you need to do it on ice. Okay, so this hot start can give you some convenience. So here, as you is shown in this gel, here you can see the non-hot start can actually give you smearing and unspecific band. Okay, but hot start, it helps to give the specific amplification and increase sensitivity and yield. Okay, also it is convenient that it can set up at the room temperature without the PCR to set up on ice. Okay. Okay, let's look into more detail on the hot start technology. As I mentioned in the previous slide, to inhibit the polymerase activity before the PCR start, we can use chemical, antibody, antibody molecule, and aptamer. Okay, so but all of them have some different effect to the PCR. So like the use of chemical to covalently link to the polymerase, it will give a more stringent inhibition to the polymerase, but because the PCR may take a longer time to activate the polymerase until it is fully active and function. Thus, this can affect the amplification of longer target or bigger than the 3 kb target. Okay, then antibody and aptamer can actually have a shorter activation time to the polymerase, but both of them are less stringent and may still result in some non-specific amplification. Okay, so the best is for the hot start is using antibody because it will have some short activation time and it do not alter the polymerase. So it allows the polymerase to act as its full enzyme activity and full function. But commercially, manufacturer, they will not tell you which technology of hot start that they are using. Okay, so when you are going to purchase a hot start polymerase, you may want to ask how sensitive is the polymerase and what is the amplicon size that can be amplified with this polymerase. Okay, next is the accuracy. The key features for PCR is the accuracy. Okay, in some of the study, we actually require a high accuracy in PCR product. We want an exactly same sequence from the DNA template. Okay, but it is common that DNA tag polymerase, it will generate error upon amplification. Okay, so that is why it's also have another activity to proofread this mistake. Okay, so DNA polymerase also have the activity to proofread this mistake. Okay, now here come the first question for today. What is the name of the proofreading activity from DNA polymerase? What is the activity name of this DNA polymerase? Okay, you can type your answer in the chat box and followed by your registration name. Okay, so we'll see who is the winner. We'll choose two winner from here and then for this first question. Okay. Okay, so here come the answer. Yes, the answer is 3' to 5' exonuclease activity. Okay, so this is the activity name of the polymerase lah. Remember in the introduction section, I mentioned that polymerase has two types of activity. One is the 5' to 3' polymerase activity. Another one is this 3' to 5' exonuclease activity, which is the proofreading function of the polymerase. Okay, so the proofreading capability of a DNA polymerase actually define fidelity, which increase the accuracy of DNA sequence replication. High fidelity DNA polymerase are enzyme with strong proofreading activity. Okay, so fidelity of a polymerase can actually be measured by using different methods, such as cloning screening assay and single sequencing and also NGS next generation sequencing. Okay, here this is with colony screening. It is often compared to a standard tag polymerase, which gives an average of 2 error per clone. On the other hand, with a high fidelity polymerase, it can actually give less than 1 error in 25 clones. And such fidelity is important for application where sequence accuracy is critical, such as in cloning or sequencing or site-directed mutagenesis. Also in some of the genetic variability study. Okay. Okay. Next, in a study, we also want to perform some of the special PCR, right? Okay, so for example, using a template that is high in GC content, or we may want to amplify a long PCR product, which may be like a 7 kb or 10 kb PCR product. Also, you may have some of the template that may carrying inhibitor like the gDNA extraction extracted from blood sample. Okay, so in this case, we will talk about the processivity of a polymerase. Okay, so processivity of a polymerase is defined as the number of nucleotide being processed in a single binding event. Okay, so DNA polymerase processivity often reflects the synthesis rate and speed as well as the affinity of its substrate. Therefore, a high processive DNA polymerase are beneficial for amplification of long template of a sequence or a template with secondary structure, high GC content, and in the presence of PCR inhibitor such as heparin or humic acid that is found in blood and plant tissue. Okay, and as you can see in this picture, high processive DNA polymerase tend to have high affinity to their substrate and incorporate more nucleotides per binding event. Okay, so thus it actually gives you a longer and complete amplification rather than a low processivity DNA polymerase. Okay, lastly, in a PCR, we will talk about the workflow for the PCR. Okay, we may want a fast workflow or a standard workflow. Okay, so we have standard workflow or direct PCR workflow, which is a fast workflow. Okay, so here is the difference of the workflow. I will discuss this in the coming slides. Now, after I have explained on the key features of your PCR, before I go further, may I ask of all of you, perhaps you already know what is the key features for your PCR? Can you write down on the chat box? Okay, so if you need your PCR to actually give you a specific amplification, you can write it as 1. Okay, if you need a high accuracy, then you may write it as 2. Okay, or you work on a special PCR, then you may write it as 3, or like you can follow with what is the special PCR that you're looking for, like long amplicon at more than 10 kb, or you need a high GC content that is for your PCR. Okay, so you can write down in the chat box so that we can understand more on the features of your PCR. Okay, so now let's move on. After you know what are the key features that you need for your PCR, it's time to choose the most suitable polymerase for your PCR. Okay, we'll start with something that is for general use. In Thermoscientific, we have DreamTech family of tech DNA polymerase. Okay, why we actually call it as DreamTech? Because it will give you the result as what you dream for. So it makes your PCR dream come true. Okay, so let's see what DreamTech can give you. Okay, unlike the tech polymerase, DreamTech is actually a modified tech polymerase which can give you higher sensitivity and greater amplicon yield, length and yield. Okay, so as referred to the gel here, it actually show the comparison of PCR sensitivity and yield with DreamTech DNA polymerase and also other commercial polymerase. Okay, it is a amplification test for a fragment with size 956 base pair using a human gDNA as a template here. So you can see here, this is the amplification for the template with the amount as low as 30 picogram. Okay, it's 30 picogram, 300, 3 nanogram and 30 nanogram. So with as low as 30 picogram, DreamTech can actually give you amplification. Okay, so it's very sensitive. Okay, next is DreamTech can also give a good amplification with high yield of long PCR. Okay, so the gel here actually showing a robust amplification of a wide range of target length with fragment size ranging from 160 up to 7.5 kb. Okay, so you can see here DreamTech polymerase can give a good amplification for product size more than 4 kb, and also the D here, the competitor also they can give you the um, amplification at 4 kb, okay, more than 4 kb. But in terms of specificity and yield, DreamTech can actually give a good yield and less unspecific amplification when the size go to bigger like 7.5 kb here. Okay, so this is the comparison. Okay, so DreamTech DNA polymerase is actually come in many different format which suit for different PCR need. In DreamTech family, there are actually standard DreamTech DNA polymerase and DreamTech hot start DNA polymerase. Okay, so for all these two format of polymerase, they can either come as a standalone polymerase only, or a master mix with all DNTP reaction buffer and polymerase. Also, it can come with polymerase and green buffer. Okay, what is green buffer? Later I will tell you. And then also master mix with green buffer. Okay, so for hot start version of DreamTech polymerase, that is actually I will explain to you. Okay, so before I go in, here is the second question for today. So what is hot start for? Remember the four features that the four key features that I explained in early section. Okay, so what is hot start for? Please type your answer in the chat box and follow by your registration name. So we will choose the two winners that answer the fastest and the correct. Okay, now the answer for hot start. Hot start is actually for increase of what? Specificity. Yes. Okay, so it is for specificity. The hot start version of DreamTech DNA polymerase can help in increase the specificity of the PCR. Okay, by looking at the comparison here, we can say that all the commercial polymerase has no problem to give specific amplification for short PCR product. Okay, but when the amplicon size become bigger, you can see that some of the polymerase will give you unspecific bands, even it is a hot start polymerase. So it actually depends on the hot start technology used for this polymerase. Okay, so however, with the special modified DreamTech hot start polymerase here, you can see the amplification are highly specific, even in amplification of big amplicon with size 5 kb. So it can give quite specific amplification with a good yield. Additionally, DreamTech Hot Start has no problem in amplified long amplicon. As shown here, it can produce up to 9 kb amplicon from human gDNA and also up to 20 kb of lambda DNA amplicon. Okay, with a very little smear and very little unspecific amplification. Okay. So DreamTech polymerase is come with own optimized PCR buffer. Every component in the buffer is well optimized to give you a superior amplification. The buffer come with pre-added of magnesium chloride, not like some of the commercial polymerase buffer, which you may need to add the magnesium chloride buffer to the buffer by your own. So you need to adjust and optimize the buffer. Okay, so this well optimized buffer for DreamTech can allow the amplification of a wider range of primer annealing temperature. And so it is not necessary to perform too much of optimization to your PCR. Okay, for example, in the gel here, you can see DreamTech can give a good and consistent amplification. Even the temperature is different from 51 degrees Celsius up to 63 degrees Celsius. Okay, and 67 is still able to give you amplification. So it can give you a wide range of temperature, annealing temperature, but it's still giving you a good amplification. Okay, and one more thing about the buffer, it can come as a clear buffer or as a green buffer. Okay, what is green buffer? Green buffer is actually the PCR buffer also. the polymerase buffer, but it actually carrying the loading die, so you can direct load your PCR product into run gel, gel electrophoresis. Okay. Also, this green buffer carrying the tracking die of the two color, blue and a yellow dye for visual tracking upon your gel electrophoresis. So let's say when your gel is running, after 5 minutes, you can see the tracking dye separation is like this. Then after 15 minutes, you can see the tracking dye. Okay, so this tracking dye allow you to visualize and make sure your gel is not running out. Okay, so what is the key features for DreamTech? What DreamTech can actually help you? It can give you a high specificity with DreamTech hot start DNA polymerase. It can give you long amplification and also with additional higher yield for PCR product. Okay, next we'll talk about polymerase with high accuracy. Under Thermoscientific, we have Fusion family for high fidelity DNA polymerase. Okay, why we call it Fusion? Yeah, because this polymerase is actually come from the fusion of reading DNA polymerase with a double-stranded DNA binding protein. So the fusion of this heterologous double-stranded DNA binding protein to the proofreading DNA polymerase is found to increase processivity without compromising catalytic activity and enzyme stability. Okay. Also, the polymerase processivity is limiting the efficiency of PCR. But in fusion enzyme, it actually exhibits a profile advantage over unmodified enzyme in the PCR amplification. Okay. So, Thermoscientific fusion high fidelity DNA polymerase was actually first introduced in year 20 year 2003, sorry, year 2003. So it provides 52 times higher accuracy than TAP DNA polymerase and 6 times more accurate than PFU DNA polymerase. Okay, since then there is actually many variation and format of the fusion enzyme was added to the product family to meet the needs of researcher. Like there is a fusion hot start DNA polymerase or fusion U DNA polymerase, which is for the UTP incorporation PCR. Okay. Okay, usually this is just a sharing. Usually researcher, they will compare the PFU DNA polymerase with fusion polymerase. So here I share a paper which compare the features of TAG, PFU and fusion polymerase using a one-step assembly of a 581 base pair long DNA sequence for protein expression in E. coli. Okay. So as you can see from the gel here, fusion polymerase can give a good single band with high intensity as compared to the other two polymerase. Okay, so in this paper, it actually mentioned that fusion high fidelity DNA polymerase, when used for DNA synthesis, has dramatically lower error rate compared to the other enzyme previously used for such application. Its fidelity is 2 to 6 times higher and approximately 50 times higher than the fidelity of PFU and TAG polymerase correspondingly. Okay, so this is actually come from the paper. Okay, so fusion DNA polymerase also come in multiple format. Here is the fusion family member. The basic is the fusion high fidelity DNA polymerase. Then there are also fusion hot-start DNA polymerase and fusion plus DNA polymerase. Okay, so for all these, you can choose for different format, standalone, master mix, standalone with the green buffer, and master mix with the green buffer. Okay, so I believe you all remember what is hot start, right? So with fusion high fidelity and hot start, it means it will give you high fidelity and also specificity. But what about fusion plus? Is it will give you double or triple in the terms of fidelity? Okay, let me tell you more on this. Okay, what is fusion plus? Fusion plus DNA polymerase has enhanced features. Okay, it actually has a new buffer formulation which allowed the DNA polymerase to work in a universal annealing temperature at the 60 degrees Celsius. Okay, so this means that the annealing temperature can be set at 60 degrees Celsius regardless the difference in the calculated temperature of the primer set. Okay, as long as the primer design is follow the general recommendation. For example, in the 2 gel here, okay, it shows the amplification of 12 targets. This gel is using universal annealing temperature of 60 degrees Celsius, while this gel is using the calculated annealing temperature. So this primer, the annealing temperature is 59. This is 59, 60, 62, up to here is this primer is the amplification using annealing temperature at 69. Okay, but here all is using 60 degrees Celsius, and you can see the comparison. There are no much different. Okay, all the amplification is very well and good yield. So with this special features of the Fusion Plus, it actually allows you to do this. Okay, let's say there is 5 different target which should amplify with 5 different primer. Okay, using 5 different annealing temperature. Now you can run all together in the same cycling protocol, which means you can actually co-cycling your PCR in one same block of thermocycler. And as shown in this gel, it will not affect any of the amplification, and it will give you what you want in the end. Okay, so co-cycling and individual cycling, the amplification is the same. Okay, so fusion plus DNA polymerase also has a very outstanding fidelity, which has over 100 times more fidelity than tag DNA polymerase. How we know this is actually in Thermoscientific, we has make a study and it determined the graph here. It show a side-by-side fidelity comparison of a few high fidelity PCR enzyme on the market by using NGS method with the molecular barcodes, and you can see Fusion Plus has a high fidelity as compared to the other. Okay, Fusion Plus also has a reformulated reaction buffer, which make it highly tolerant to PCR inhibitor. So thus, if you are working on sample with inhibitor, like blood or stool samples, Fusion Plus will work best for your PCR. Here is an example that Fusion Plus DNA polymerase is working well in PCR for sample that have inhibitor, like the humic acid, hemine, and xylon from different sample type, from plant, soil, and also from blood. Okay, so the amplification is stable and consistent, and it can give you a high yield amplification. Okay, Fusion Plus also have the high efficient in GC rich amplification. Okay, so this just is show you 16 target with high GC content, with different high GC content, different GC content here. Okay, so this is the percentage of the GC content, and then this product were actually amplified from 15 nanogram of human gDNA, and you can see that Fusion Plus can amplify very well and produce PCR product with very high yield. Even the GC content is as high as 80%. Okay. Okay, so now what is the key features for Fusion high fidelity DNA polymerase? First, it can give you high specificity with the hot start version of Fusion DNA polymerase. Then, it also give you high accuracy with 100 times more fidelity than tag DNA polymerase. And it is a special formulated buffer, can give you a good amplification for high GC rich template, long template, and also sample with inhibitor. And additionally, Fusion Plus can allow you to co-cycle all your PCR with different primer set under the same PCR protocol with annealing temperature, universal annealing temperature at 60 degrees Celsius. Okay, lastly, if you need something to shorten your PCR workflow, we have the direct Pcr kit from Thermoscientific. Okay, so in standard PCR workflow, it will usually involve sample collection, and then you need to isolate the DNA and quantify the gDNA, check all the yield, and before you can proceed to PCR, right? But to determine a better speed in the study, we can actually shorten this workflow by skip the front part of the template preparation and go direct to the direct PCR by using the Thermo Scientific direct PCR kit. Okay, so it can skip all this method to sample collection and direct PCR. There are many advantages in using direct PCR. One of the main advantage is there is no need for any additional nucleic acid extraction step, so it's actually save your time and your money for buying the DNA extraction kit. Okay, also it uh, it also very much convenience with fewer pipetting steps, so less prone to pipetting errors also. So Thermo Scientific Direct PCR Kit also come in a master mix format with pre-added loading die for direct gel loading following the PCR. So all these direct PCR kit are also compatible with a wide range of sample type, including human, animal and plant sample, and it requires only a very small amount of sample material. Okay, here are the family members of Thermoscientific Direct Pcr Kit. The Fire Plant Direct Pcr Master Mix is actually designed for plant tissue. It can extract from leaf, root, seed, and also it works great for cell with a tough cell wall, such as fungi uh, or gram-negative and gram-positive bacteria is also can work. Okay. Then next is the Thermoscientific Fire Tissue Direct PCR Master Mix. So this master mix is designed for various type of tissue, including hair, nail, skin, and even it can extract from saliva and buccal swab, muscle, feather, fin, and also scale. So this can also be from various of the source, like insect, worm, Drosophila, or uh, FFPE also. Okay. Okay. Lastly, we have the Thermoscientific Fusion Blood Direct PCR Master Mix. Okay, so this master mix is good to extract from human and animal blood. Okay, so the blood can come in various type, various storage condition, like fresh one, frozen, or stored in citrate, heparin, EDTA, or FDA cut, or Whitman. Okay, cut. Okay, there is actually two protocol to choose from the uh, Thermoscientific Direct PCR. So there is a direct protocol, which there is a minimum hands-on time needed, because the sample, such as in this case punch out of the leaf tissue, can simply add to the PCR master mix, and this reaction mix can then go straight to PCR, which can be complete in 30 minutes. Okay, although this is a very the fastest protocol, the drawback of this method is it only uh, allowed one reaction to be performed, and the genomic DNA cannot be archived. So if genomic DNA archival is required, perhaps a multiple PCR reaction or a long or difficult amplicon is needed. Then the dilution and storage protocol is better to be used. Okay, so in this protocol, actually the sample will be first added to a dilution and storage buffer, which contain reagent to release the DNA. Okay, a small portion of the supernatant containing the released gDNA here is then input into the PCR master mix for PCR. And in this way, the genomic DNA can be used for multiple time or multiple PCR reaction. Okay, so this protocol is a bit longer than this one. So the direct protocol is still the most fastest. Okay. Okay, so what is the key features for Thermoscientific Direct PCR Kit? Certainly, it can give you a fast workflow, and it is suitable for various sample type, but with a drawback: if you need the gDNA for more downstream work, then this kit may not suitable for your study. Okay, so you still need to perform gDNA extraction, right? Okay, now here come the third question for today. Thermoscientific Direct PCR Kit is suitable for which sample type? There are three sample type, right? which I mentioned, there is actually 3 kits for 3 different sample types. So what is it? You just need to write down your answer at the chat box, followed by your registration name, and we'll choose 2 winners who answer the fastest and give the correct answer. Okay, so what is the 3 kit or 3 direct PCR kit with 3 different sample type? Okay, the answer should be 3. Yeah. Okay, so what is the answer? The answer is plant, tissue, and blood direct PCR kit. That is kit for plant and kit for tissue and also for blood. Okay. So now you should already know which polymerase to choose for your PCR, right? As I have mentioned so many. So here I summary it for you according to some of the application. Okay, so if you are doing standard PCR just to verify the gene or just to confirm the presence of gene, then you can use a dream tag. Dream tag is suitable for general application, and it is good to give consistent and high amplification yield. Also, it is able to generate up to 7 kb PCR product. And if you are working on PCR that need a high specificity like genotyping or multiplex PCR or nested PCR, DreamTech Hot Start is good to give the specific amplification, and it allows you to set up the reaction mix in the room temperature. Okay, so next is for study that need high fidelity PCR like cloning, protein expression or mutagenesis or genotyping study. Fusion high fidelity DNA polymerase or the hot start version of the fusion high fidelity DNA polymerase will be suitable. So it is also suitable for automation. Okay, next, if you are working on some special PCR like GC, high GC template, or your sample may carry some inhibitor like blood sample, it is recommended to use Fusion Plus DNA polymerase. Okay, Fusion Plus is also suitable for co-cycling. So if you have many primers and many samples to work with, Fusion Plus will be a good choice for you because you can actually co-cycle all the PCR reaction under the same protocol and same thermocycler block in one thermocycling. Okay, so fusion series of the polymerase is also have another type of polymerase like the fusion U DNA polymerase. It is actually specially for the study on the template with uracil and need DUTP incomplete PCR. Okay. Okay. Lastly, if you wish to have a short PCR workflow, you can consider on Thermoscientific Fire Direct PCR Master Mix. Okay, that is for plant, tissue, and blood direct PCR Master Mix. It can save your time and it works in many wide range of sample type. Okay, and it is suitable for DNA barcoding, which you actually just need to add the sample into the and run the PCR. Okay. Okay. So now next is if you have start your PCR, then you actually find some difficulty in determine the result that you want. Here I want to share some of the common PCR difficulty that is face that you maybe you will face it also. Okay, So one common question is, how do I eliminate non-specific band in my PCR? Okay, your PCR have a non-specific band. Okay, so as I shared previously, polymerase activity prior to the start of the PCR cycling can actually lead to non-specific band. So an easy way to overcome this is to choose a hot start DNA polymerase. Okay, if that does not work, then you can also try to increase the annealing temperature or decrease the annealing time to reduce the non-specific binding of the primer. Also, it is important to ensure your primer do not have the self-complementary sequence at the 3' end, which can actually lead to primer dimer and reduce the yield. Okay. Also, you can try to lowering down the magnesium concentration as the magnesium stabilize the primer binding, even for the non-specific one. And lastly, a touchdown and nested PCR are the modified PCR method that can be designed and used to increase the PCR specificity. Okay, so next, if you are experiencing smearing on your gel electrophoresis after the PCR, first eliminate the possibility of contamination by using a negative or a no template control. So when you make sure there is no amplification in your negative control, and also you need to ensure your uh, gel is precast or cast properly, then you need to go for the other option. Okay, so very often an intense background smear is due to overcycling. This is caused by using too much of template or enzyme or primer or DNTP or too much of cycle. So try to lower the annealing temperature, and also you can try to reduce the non-specific product by optimize the annealing temperature and the extension time. Okay, and it's important to ensure that your primer are well designed, using a primer tools to check and for the property of the primer. And then also you need to ensure your primer is not too old or degraded. Okay, so primer storage, it will be better to store at to resuspend in TE buffer, which can uh, preserve your primer more better. And then the storage, it should be up to uh, 2 years around that. Okay, it will be stable, but after that maybe you may want to like order a new one after 2 years. Okay. Okay, so the opposite of smearing problem is having the low PCR product yield or no amplification at all. So this will require to look into several PCR conditions to optimize. First, there are is there any PCR inhibitor that can inhibit the DNA polymerase, such as high concentration of uh, sodium chloride or KCl or DMSO or amine in the blood sample. So check whether there is possible of PCR inhibitor. If there is, try increase the amount of DNA polymerase or increase the PCR volume to dilute out the inhibitor. Okay. Secondly, look into optimizing the annealing temperature. Um, okay, by using you can actually use the magnesium correct concentration to optimize. Also the primer design, and also you need to ensure there is sufficient denaturation and elongation time. Okay, so optimizing the PCR is very important. And if your template actually contain high GC region, so you need to choose the DNA polymerase which suitable and with optimized buffer for GC rich PCR. Okay, so with this I end my slide here, and thank you. If you have any questions, do type your questions in the chat box. We will try our best to answer you.